one day, some set of people will bring solutions to these problems that is causing problem in the world. And many of us, we have formal education. We have BSc, we graduated from this so department, and yet we are not even thinking that okay, I can actually make an impact when it comes to the world of science. And it's not our fault most of the time. It is because many of us, we don't have everything necessary to do all these things. And that is why we need computational power. We need computer technology to maneuver our way, to see what we can do, to see some of the discoveries we can actually make in the world of science. So now I'll be moving to the next slide now. When it comes to drug discovery, it is actually aimed at developing a solution for a particular disease. Take, for instance, when COVID came, people were running it and scatter. What will happen? Where are we going to get solutions to this? Where are we going to provide this that can kill COVID? And a lot of people were dying. And if we begin to think on, when are the scientists going to have, all the required resources and everything to provide a solution to this COVID problem, it will take a whole lot of time. And this is where computational chemistry comes in. This is where computational technology comes in to design drug, to screen many drugs. If you look at my slide, I just added some of these things so that you can read as well and follow me through. So when we say drug discovery, is a process in which we go about looking for solutions to some of the problems that is ravaging the world. We still have so many diseases today that yet they don't have a cure, and hence we need scientists who can help us. Then when it comes to drug design as well, when we design a drug, there are so many things we need to evaluate before we roll out drugs to the society. I tell you, some of these drugs we are using today, maybe like the anti-malaria and all these things we make it, before they actually got the approval, it takes a whole lot of years. And with the help of computational chemistry, computational biology, I tell you, we won't spend the amount of years we spend in getting to the approval stage. When COVID came in, then now we need to screen drugs that can actually work for COVID. We need to develop a new drug that can cure COVID. But then we don't have the resources in our environment. Take, for instance, many of us we are in southwestern part of the country. Some may actually be in the northern, whichever place. How many research lab do you have in your environment that can actually give you some leverage to see what you can do about some of the diseases that you are having around us? Where will you get the isolate of some of these diseases to check if this compound can work for it, if that compound will not work? But with the help of computer technology, we can actually screen many drugs against many diseases targets. In COVID-19, permit me, I'll be making use of COVID-19. When COVID-19 started, the researchers were running, okay, let's look at some of its mechanism of operation, how how human cell gets infected, and how it was transferred, and many other things like that. So now a time came whereby the scientists identified some of the some of the protein targets in COVID. COVID actually has some proteins in it that helps its operation. There's a particular protein we call mean protease, and we also have another one called papain protease. You see, these two proteins, they help COVID in attaching itself to the protein, to the host cell. So now we need to develop drugs that can actually inhibit the activity of those proteins. And for us to do this, we don't really need to visit laboratories. We don't need to spend time isolating different compounds from many, plant extracts we're having around us. Many of us, we did in our project or in any research we have done, we did some work on some plants or maybe some food that we just okay, this food is good, can cure this. It is not enough to say bitter leaf is good or is whatever for this particular condition. We need to know the bioactive compound that are present in them. I hope we are following. Are we together?
Yes, sir.
Okay, thank you very much. I appreciate that. So we need to exploit some of these processes to see how we can, in our little environment, work on something and come up with substantial, some, proofs that, okay, I can say this because of this evidence I have. Many of us now, we just...
Right on, man of God, right on.
Okay, thank you, sir. Many of us now, we just have, maybe when we have pie, and the next thing we do is, let me go and find a wuro, let me go and find bitter leaf. But as a scientist, do you have proof that bitter leaves actually works for this thing? But with computational technology, with computer technology, we can actually explore this. Recently, there was a time I was looking at some protein targets in COVID. Then I also screened a reference drug, which is common, and I compared it with some of the compound. I feel like, okay, this could actually work, and I kind of see some similarities between these drugs. That, okay, this compound has some similarity with some of the already approved results, already approved drugs. So this is where computational technology comes in. So let's follow on. We have so many things. I will actually walk us through a particular practical session. I just want you to follow and do that as at when necessary. This thing is not is a techniques everybody needs to have, because we are in the world of technology now. Everything you just need to do here is you need a good computer and access to so many things, and you begin to explore so many things. This skill, I tell you, many even lecturers who are having PhD or professor, they don't have access to this thing. Not because they cannot, but they are kind of old. For some of them to sit down and begin to grab some of these things, it will take time. So let's move to the next slide. In developing drug, there are two approaches that we usually follow. Number one is the traditional approach, which is the old method of developing drugs. And how do we do this? We kind of isolate uh, probably a particular compound from a plant extract, or we isolate from different sources. Then we take them to the wet lab and we isolate our organisms as well. And come in, let me, okay. Okay, so we isolate our organism of interest as well that we want to really work on. And we subject them to various processes and we look at it. In fact, sometimes during some of these processes, a lot of things can happen. The organism you're actually working on can actually mutate. Before you know, the drug you are thinking could actually work for it will no longer work for it, thereby setting all your research effort back. And sometimes probably the compound you are trying to, I hope you can see my screen. I'm actually in a screen whereby we have traditional approach. Can we see it?
No.
Oh. How about now?
Yes.
Okay. So maybe we'll just use it like this. So this traditional method, this traditional approach could take years, could take many years before we arrive at this target. But with modern approach, one, it is fast. With the aid of computer technology, it is very fast. With the aid of some computational tools, it will reduce the years in which we take to discover drugs. Now, when COVID came in, please just permit me, I'll be using COVID now. A lot of people were in fear, quite number, whether we believe it or we think it is just one propaganda, but whatever, that we are not concerned with that. When COVID came, a lot of people were infected with this virus. We need some drugs to actually keep people pending the time other drugs will be developed. If scientists approach the traditional method only, I tell you, they will not be able, many people will die. The scientists, what they did was that they adopted the modern approach. And the modern approach is to make use of computer to screen some drugs. Mr. Yemi will walk you through some sequence and everything like that. So what they need to do is to screen some compounds. We actually have a database. I'll still show you when we get to that section. We have a database whereby we have different compounds that have been isolated from different plants. After this training, I will encourage you, most especially those of us that have worked on plants, take your plant, go to internet, go and search for the bioactive compound in that plant. It is not enough to say that moringa leaf is good for this. Those are normal, you can't present that in a standard setting. So there are some compounds that are actually very, very, active in that plant. So we have a database where we have all these compounds that have been, some scientists have actually worked on them, and there's a way they crystallize them, and they have them. We have access to them through our computer. We can actually screen these compounds against the disease we are looking at. I said the other time that COVID-19 has two important proteins, main protease and papain protease. COVID uses one of the two to attach itself to a particular receptor. I will show you some of those things. I just hope I have more time to do that. COVID uses one of those proteins to attach itself to the host cell. So once attachment is done, I tell you, you all know the subsequent reaction that will take place. It will inject its genome into the host cell before replications here and there. So we need a drug that can actually stop that can actually stop the activity of those proteins on the surface of COVID. People with HIV now currently we don't really have a drug that can cure HIV, but we have drugs that can actually reduce the rate of replication of the virus in the body system. That means scientists were able to come up with drugs that can inhibit the activity of the replication activity of HIV. So then, this is where computational science comes in. We need technology to screen some of these things. Not that we need to go and carry HIV in the isolation center, we begin to culture it, we begin to monitor it, and begin to apply this and that. All those, process will take us a long time. We are not saying those processes, they are not useful. They are of course good, but it takes time. They are very expensive. Imagine you want to take an isolate of COVID-19, a particular strain of COVID-19, because we have different strains. You know how much it will cost you? You will take a lot of approvals from so many. In fact, they can't even hand it over to you. But I can tell you, with your computer, you can work on COVID. I have done that, and even more of protein targets like that. So now we have these drug design strategies. We usually, most of the time, like I said earlier, we have high throughput screening, whereby we'll be physically testing large number of compounds, often in millions, against a particular biological target using automated assays. We all know all those assays in our laboratory, but we call something virtual screening. This is where computational simulation of ligand to protein interaction. This is a method in which we make use of the information we retrieve from all these databases, and we retrieve protein targets from some other databases as well, and we screen them and see if there is actually some binding. We call something binding affinity. We also call something docking score. So when a particular a particular compound has a good binding affinity, it has a good Dawkins score. We can actually look at this thing that this particular compound could actually cure this. Meanwhile, we've not even taken them to the lab. This method will save us stress of going about, getting leaf from South Africa, getting leaf from China. I tell you, you can work on leaves, plants from other countries with computer technology. I hope we are together, please. I just want us to have this understanding so that it will drive you after this time on to seek to know more. All these things are not, they are not magic. They are not. They are just computer technology. I can remember when COVID came, we were just sitting there, to everybody listening to news when they will announce that we should go out. Everybody is locked down, and we are not contributing, and yet we are in school. So now, when we actually discover a particular drug, when we discover a drug, when we discover a particular compound that this compound could help, there are various processes we need to subject them to. There are various places we need to take them to. In fact, before a drug gets to the market, it has to get some approval. It all depends on standards set by each countries. I know Nigeria has some standard as well, and in America they also have their standard as well. So in an instance, maybe you worked on a plant, let's say bitter leaf, and you did some computational analysis and you looked at it that, this compound could actually be good. This compound, or I isolated this compound from these two sources, and I found out that quite number of bioactive compound in it were able to react, were able to interact with the protein target of this disease. Henceforth, what do we do? Now, maybe you are the type that you make use of a bitter leaf, and you have your friend who actually worked on maybe moringa leaf, and you are just thinking that, how can I actually check the efficacy of this thing? Sorry, I'm also trying to do something here. Okay, how can I ensure that this thing is actually working? When you work in the lab, you also have there's something we call in silico model. I want us to take note of that. Just like we have in vivo and in vitro, how we all understand that. We also have something we call in silico model. That is, this is a method in which all our, research, everything, probably we are working on a disease with all the plants we want to use against it. Everything is done using computer technology. That is what we call in silico model. It's some of the new developments we are having in our world today. So once we're able to identify this compound, that this compound is good, then we need to go through some processes. Now, we need to check the preclinical testing. Before we get to this stage, that virtual in silico model will actually assist us, will actually help us to screen many compounds, not just limited to our immediate environment. We screen many plants from different continents of the world. Then we now see the ones that are good. We remove the ones that are not good. And these are the ones we now, let's see what these ones can do. Let's see what this one can do in the wet lab. Let's see what this one can do in this particular, rat, or maybe you want to use Drosophila or anything you want to use as at your preclinical stage. So we follow these processes. And there's another important thing I have here. We call it pharmacokinetic studies. We need to understand it is not enough to say this compound is good. What about the side effects? This is where all these people selling herbs, where they don't really, understand. And as a scientist, we shouldn't, follow the multitude and just say, okay, our forefathers have been using it, let's make use of it. Yes, it may actually be good, but some can actually cause some damages to your body system. I'm going to walk you through a process to to actually check for that. Then we round up this section. So that we also have toxicity studies. You see, all these preclinical stages, you can actually do everything with your computer. It is when you now get to clinical trials, okay, let's see, let's assess this drug that has proven to be good in our virtual screening. Let's test them on maybe a rat. Let's see how it will do. Let's confirm, because it's not enough to just say one method is good and you are not subjected, because in approval, before you take approval, you must have tried various means. That's why when before a drug can be released to the society, we see that during the clinical trials, they bring people from different country, different backgrounds, and check, because we have, some of us have different, genetic information. Some may develop allergy to a particular thing and some may not take. So they begin to evaluate every possible thing that could cause problem. So we get to these clinical trials, we check the phase one, we give them a particular low dose, we check how that is working with them, we check the phase two and phase three. But we are not here for that. We are just looking at how we can use our computer technology to exploit some of these compounds we're having around us. Like I said earlier, virtual screening of drug, we have give you the opportunity to screen thousands, millions, billions of compounds in the world. I read a research that when COVID came, the scientists screened 6,500 compounds. They found out that six actually had some, fantastic ability to interact with COVID. And they took these six, they did some other further analysis. Before you know, they came out with some results. There's a particular, some people are saying chloroquine was good for COVID. They don't just wake up one day and say chloroquine is good. They did a kind of virtual screening. They did a kind of analysis to arrive at that. So now, how virtual screening shapes the fight against diseases. We'll be able, with virtual screening, traditional drug discovery method, which is very slow and expensive, becomes a thing of the past. We can exploit libraries. I told you we have databases whereby you can take compounds isolated from this plant and use it for the disease that is causing problem all around the world. Then when you come up with some initiative that, this is this thing, then you begin to talk about getting your research discovery into the scientific world. Now another thing is, there is targeted precision. I love that. We call something targeted precision. It is, all these organisms, they are very, very wise. How do I mean? Some of them, there's something called antimicrobial resistance. There's a mechanism in which some of these organisms adopt to evade some of the, things you give to them to kill them. But in virtual screening, that is in silico model, you know where you are going. Just like COVID has RNA, it has some other things in its system, but we just need the one that COVID uses to replicate, which is either main protease or papain protease. Once we're able to get that off, I tell you, we can as well reduce some of the symptoms COVID is causing the body system. Let that person mute so that you don't disturb others. Please, you can mute. So now, I go to the next slide because my time is kind of going. So, what are the impacts virtual screening and development of drug, has offered us? One, we have optimized lead compounds. There's something we call repurposed drugs. Some drugs were repurposed. There's a particular drug they used in the treatment of COVID. Initially, it was used for hepatitis. I think hepatitis B. I think remdesivir or something like that. It was initially used for hepatitis, but with the use of computer technology, the scientists were able to discover when they were screening drugs that, oh, this particular drug can actually do something, can actually, reduce or stop the activity of COVID. Some countries gave that drug to COVID patients, and there were some interesting results they were able to obtain. Pending the time other drugs will be developed. A time came, a particular country in Africa said there is a plant that actually good, that they okay, just take this tonic and do this. Scientists, the world, the WHO will not be able to take that because it is not enough to say this. I don't want to. Should I call it concussion? Can actually work. What are some of the scientific backgrounds, scientific research you have for this? Can you actually convince us with this? So this is where we need more scientists, more people to venture into this, most especially in Africa. If you look at Nigeria, as big as Nigeria is, as our population is, we have two universities that offer bioinformatics as a MSc program. And I think that is Covenant University and University of Ibadan. And that means, I can tell you, that means if that course is limited to two universities in the Nigerian system for MSc, then what is happening in other universities? Are we going to stay with the normal traditional method of screening drugs here and there, and before you know, we won't be able to achieve anything? In fact, some of the isolates we are having, they have already undergone mutation, and we don't even know, and we are trying to just see that things are not working. Do we have all the properties, the resources to carry out proper traditional, approach of drug screening? And that's why we are, in Africa, limited. We're always depending when we in USA come with their something. So now okay, yeah. There's another thing: reduce animal testing by predicting potential toxicity. Now we're going to do that this afternoon before we round up this session, to check for the toxicity of a particular plant. Some plants can be good, but they are toxic. They can cause liver damage. The liver will not be able to handle them. So what's the benefit if I take a particular drug to cure this, and before you know, the person already has liver damage? And that's what happens in many people that uses all these herbal products. Before you know, they are already having problems. This person that was okay sometimes ago, before you know, already has liver damage. They are being diagnosed of different diseases. It is because there is no toxicity test. And even after this time out, you can, pick up a plant around you that people have been saying. Is anybody saying anything? Please, let's mute our mics. So after this time out, you can exploit plants around you. Go to the internet, retrieve some of the bioactive compounds that are present in them. Check the toxicity test yourself. Look at it and make some findings, and you get more understanding of some of these things. Probably the artemeter you use at home, you just feel like, artemeter, I'm just using artemeter. What do I even know about artemeter? So, and also we have personalized medicine, CADD. We call it computer-aided drug design tools. Can actually help us design drugs tailored to individuals, genetic makeup. Mr. Emi will walk us through that. The genetic makeup of an individual. Would this drug be okay for this person? Now, I think the complete genome of human has been sequenced. I think something like that. With that, we can actually exploit so many things. CADD, computer-aided drug design, rather, computer-aided drug design. So please, let's take note of that for this disease and that. Now, take for instance, you have bitter leaf, which is we all know as ewuro. So, and you just feel like, okay, what are some of the bioactive compounds that are present in it? What you just need to do is go here, search bioactive compounds in bitter leaf. So when you search this, you have some. So when you need some of the bioactive compounds in bitter leaf, you just do a simple Google search, bioactive compounds in bitter leaf. You search. Then when you begin to open, you begin to see some of these things. You begin to see compounds like alkaloids, saponins, terpenes, steroids. But there's an important thing here. It is not enough to just conclude what type of flavonoids, what type of steroids. These ones are just like compound, name. We have different types of flavonoids. So when you now click this particular document, that's when you begin to see other compounds. Like you begin to see compounds like quercetin, and you begin to see different compounds that are different from all these compound name like flavonoid. We have different alkaloids. So when you now take this, it is these compounds you retrieve, then you begin to work with. Maybe some other times. I think my time is fast spent now, and I need to hand over this session. I would have walk us through a particular thing, but then you can do some background study about the plant you are working on and find out what you can actually do with that plant. So I would like to hand over now so that we can have the next session we are having on board. So, Miss Precious.
Yes, thank you so much, Mr. Shiu, for that wonderful presentation. That was awesome. And I hope you all learned something from his presentation. We will
Yes, we
have the
can hear you.
next presenter by the name Mr. Adeyemi Sunday. Mr. Adeyemi Sunday, you have the floor.
And once again, I want to say a very big thanks to Mr. Shion. Thank you very much for coming around. And then for all that have joined this call, thank you, everyone. Well, I should have come first, but due to some managerial duties, that was why I couldn't start the webinar with us. Yes, as Ms. Precious has announced me, my name is Yemi, and by the grace of God, I'm a student of bioinformatics, and then we have been able to do one or two things with bioinformatics. The aim of this webinar is to bring bioinformatics down to grassroots because we understand the fact that many of our indigenous universities, they are not well grounded when it comes to computational biology. And that is why we have taken it upon ourselves as an institute to bring bioinformatics to you, to everyone. So I will be doing the introductory part. Mr. Shen has rightly done justice to so many things. Biological database. That is why we call it biological database. This one is different from mathematical database. This database has to do with biological information. Okay, so types of bioinformatics database. We have three types of bioinformatics database. Number one, we have the primary database. As you can see, example of primary database is the gene bank, the DDBGB, DDB, WDBG, PDB, cis protein, PIL, TREM. So primary database in layman, this is database that comprises an original biological information submitted by researcher. So we call it archival database because the information you will be seeing on primary database have not been you know have not been manipulated. These are the information directly from the source. Let me use that word. So, for instance, now you as a researcher, you are working on a particular DNA sequence. You get your sequence, then you submit on primary database because this sequence has not been worked upon by another person, another researcher. So, and then the next one is secondary database. Example as inter, prob, uniprot. Yes, we'll briefly look at some of those things later. So, secondary databases comes with information that has been manipulated. Primary information that have been manipulated, that have been, worked upon by another researcher, by another, bioinformatician. So, example is inter, pro, and unipro. Then the last one is the composite database. Yeah, this composite database, as the word implies, comprises primary database and secondary database. Example is SysProp, TREM, OWL. So these are the biological database that we have. And these three biological database can be categorized into two: the generalized and the specific database. The generalized database, these are database that comprises all biological information. You're looking about DNA, RNA, protein, you will see it there. A typical example is NCBI, yeah, as we'll be looking at briefly. NCBI, so it's a generalized database. Then for the specific database, you are working on protein. You can see, nucleotide sequences in a specific database. Example is Uniprot. Yeah, Uniprot is a specific database that has to do with protein information. So I believe you are getting the two description now of the databases that we have. So now sequence analysis. What is sequence analysis? Sequence analysis is the process of subjecting DNA, RNA, peptide sequence to any wide range of analytical method. Yeah, you want to analyze your sequence, either protein, either RNA or peptide sequence. So you want to analyze them for a particular feature, function, and structure, or you want to look at the evolutionary trend of that particular sequence. And then one aspect of sequence analysis that we'll be looking at this evening is sequence alignment. Now, what is sequence alignment? Sequence alignment is the procedure of aligning two or more sequences together. As you can see on my slide, one is pairwise alignment, that is, you are aligning two. Now, let's quickly look at the type of alignment because of our time. There are two major types of alignment: the pairwise alignment and the multiple alignment. What is pairwise alignment? Pairwise alignment, as its name implies, aligning two sequences together. You are aligning two sequences together. Either you want to look at the function, you want to look at the structure, just two sequences. Then for multiple alignment, more than two sequences, probably sequences from E. coli, sequences from Vibrio, sequences from other organisms. You want to align them together, so that is multiple alignment. As you can see here, and then this sequence alignment can be as well categorized into three types of sequences as well. We have, sorry, I will go back to the previous slide, but I want to explain this slide: global alignment and the local alignment. What is global alignment? Global alignment is the end-to-end alignment. You are aligning all the sequences. Then for local alignment, as the word entails, just a local region. Probably you are working on a particular local region on the whole DNA, on the whole RNA. You will be aligning. So this is example of the first one here. A is the global alignment. Can you see all the sequences are aligned? This is the first sequence on the top, and then the second sequence, all the sequences are aligned. Then we have the semi global alignment. Though this one is not very majorly categorized, but some articles you will see semi global alignment. Look at this: aligning not all the sequences, but at least two-third of the sequence together. Look at it now. Omitting the first sequences here, then omitting the last sequences. So aligning these regions together. Then for local alignment, you are aligning a particular local region. Probably this particular local region you are working on, this particular sequence you are working on. So you just align the local region together. So I've explained this now. Two for sequence alignment now. For please, this is where you have to have your pen because we'll be doing some two for sequence alignment. We have the emboss need for pairwise alignment. If you want to align for two sequences, we have the two emboss, which is emboss needle and emboss water. We'll do that shortly. This is the link. This is the link on the. So the next one is for blast. Yes, NCBI has this. What is blast? Basic local alignment search tool. Basic local alignment search tool. Then we have the line. This example of tools that are aligned for pairwise alignment. Then for multiple alignment, we have muzzle. I used this muzzle some few weeks ago to align about 30 sequences. Yeah, T coffin. Yeah, mapped cluster W. I use cluster W as well. Then mega. This is one of my favorite tool when it comes to sequence alignment. So that is the will be the end of the introductory aspect now. So we want to quickly do some hands-on training. I believe quickly do some hands-on training on the sequence alignment and then stuff you need to know. I believe you are all with me. Are you all with me?
Yeah, with you, sir.
Yes, sir. Yes,
Okay.
sir. We're with you.
Yes,
Yeah,
sir.
let me end the let me end the okay. So I'm still sharing my screen. I'm still sharing my screen. I believe you can see. So for sequence alignment, I said it earlier on that when aligning sequence, the first thing you must do is how to retrieve sequence. And I've walked you through the type of database that we have. If you want to align sequences, you must retrieve your sequence. And don't forget, when I was defining database, I told you that it is an online archive in which sequences are being retrieved. You manipulate sequences there. So now for retrieving of sequence, which one should we retrieve? Is it RNA, DNA, protein that we should use as example of our hands-on training this evening? Which sequence? Okay, let's go with the. I know I have some. Okay, protein. I know there's some by about chemistry now. I mean, so many of them here, so I know they like protein. So let's use protein. Now, I told you that protein now. If you want to retrieve protein sequence now, I've walked you through some of the database you are going to be retrieving that. You can retrieve it from the generalized database. I told you that generalized database comprises of all the biological information. You will see RNA, you will see DNA, you will see protein, other stuff as well. You will see structures, so many things you will see there. But from specialized database like protein now, yeah, as you protein now, just uniprot, yeah, you will see protein there. So, but then I do encourage I do encourage people using a generalized database because when you are doing the analysis, you won't have any issue at all. So let's use a generalized database. Let me open. So NCBI, just type NCBI, National Center for Biotechnology Information. You can see my screen. You can see my screen. Just click on NCBI. You can see. Are you still seeing my screen? I believe you are still seeing my screen. Please
We can
unmute your
see.
mic. Okay, thank you, thank you. So this
You can see us, sir.
NCBI is okay, thank you, sir. This NCBI is a generalized database. As you can see, you just move the cursor to where you have all databases. I told you that is a generalized database. You click on it, you can see that assembly, all that, blah 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 blah. You see nucleotide, you see peptide, you see protein. But since we are working on protein, let you click on protein. So the protein sequence you are working on, either probably from E. coli, um, Shigella, other stuff you are working on. But then, can you just suggest which one should we retrieve this evening? Which one should we retrieve? we want to do sequence alignment, we want to do the pairwise alignment, but for the multiple alignment, we won't be doing that on this call because of the time. Because using to make it more specific, let's type E. coli anti antimicrobial 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 resistance. To make it more specific, resistant gene. See, okay. Look at it now. Can you see we have the TLB family total resistant protein partial for Escherichia coli. These are so many sequences. These are so sequences that have been submitted by researchers that is causing antimicrobial resistance. So let's try to align the first and the second sequence together now. We are aligning this sequence together, and let's see the either mutation has occurred or what happened. Now we are going to download in faster format. Don't forget we have two type of format. When we get to a bit of advanced bioinformatics, I will let you know the more of the two type of format that we have. We have two type of format: the faster format and the fast queue format. For the faster format, can you see for the faster format? You just click on the faster format here. Click on it. The faster format. One of the differences between faster format and fast queue format is that for faster format, the sequence will be annotated starting with a greater than sign. As you can see the greater than sign here, I believe you are seeing the cursor. I
Can
believe you can see the okay, you can see the direction of my cursor. Okay. The direction of
can
my cursor here. Okay, yeah. Look at it. It started with a greater than sign. WP. These are the annotation in which this sequence is being submitted. So this is the sequence now. So that is the faster format sequence. Now, for you to align two sequence, one major tool I use for aligning sequence is emboss. Emboss needle. We have two type of emboss water and emboss. Look at emboss needle for pairwise sequence alignment. Pairwise sequence alignment. Okay. So sorry. So can you see the my time is gradually coming to an end. So can you see this is where you will paste the first sequence here. The first square will be pasted here. Then the second sequence will be pasted here. Then you run the analysis. You submit the analysis. So excuse me. The first sequence now. Let's paste the first. You just highlight it, copy it. Can you see this as you have to set the function of this tool that you are working with. If you are aligning a protein sequence or a DNA sequence, either protein sequence you are aligning, but because we are aligning a protein sequence, we will click on our protein sequence, then you enter. Can you see please enter the faster format? Is the faster. Please you have to be careful so that you will not delete any sequence. You will not delete any amino acid, because if you delete it, it will affect uh, the result, the output. So I said we are aligning the two sequences my time. So let's retrieve. This is what we call retrieval of sequences. We are retrieving sequences now. So let's align. This one has an amino acid. Now, what is the goal of aligning these sequences? One of the goals is uh, to look or to check if mutation has happened. These are resistance gene. These are different resistant gene of E. coli. So sequence alignment helps us to understand this particular gene very well. So after input, then you don't change. This is a default setting, so you don't have to change anything. So to be notified by email, you will just click on to be notified by email. Now, if you want the result to be submitted to your email, so you just input your email. Okay, my email, my email. I'm inputting my email so that so then for the title, you can as well put title antimicrobial, antimicrobial resistant, say E. coli. Just put any title, then you submit. Let's await the uh, okay. Look at the announcement on this one. We will be retiring this page on the 31st of January 2014. That means that whichever work you want to do with MBOS, this particular information has been keeping since last year, even the last time I used MBOS. So on forward from 31st of January, you will not be able to access this particular tool. Otherwise, then your job has been submitted to your email, and don't forget that the result will just be available for just 7 days. So await. Let's see the result of our result. Let's see the result of our work. This is the result of the alignment. I will explain. This is the result of the alignment. Can you see that we align two sequences together? Two sequences, but this is the first sequence. Can you see that there are so many gaps here? Now, for the similarity, identity. Let me put the cursor there. The length of the whole sequences is 111. Then for the identity, for the similarity, look at the gaps and the score. The score is just 21.0%. I will explain all these things. Similarity and identity, well, they are the same and then they are not the same. I believe you understand that one. They are the same. You will see some researcher, they will tell you that similarity and identity are the same, and then others will tell you they're not the same. As you can all see, look at the score. This one is 9.9%. This one is 18.0%. You can say, what is identity? Now, it's telling you that the two sequences, there are two antimicrobial sequences that are causing antimicrobial resistance. Now, the identity is just 9.9%. That means the two sequences are not identical. This one can tell you that mutation has happened on this particular length. So look at the similarity. Now the similarity is telling you that this particular two sequence are they from the same strain. We have so many strains of E. coli that causes antimicrobial resistance. Are they from the same strain, either E. coli 0.017, E. coli, all those E. coli you get? So, are they from the same strain? How are they similar? And you can see that it is just 18.0% similar. Then look at the gap. This gap is telling you that the deletion that has happened on this sequence, the deletions that are happening on this sequence, you can see from first amino acid to almost, this is 50 sequence amino acid to almost this, but let's say something around let's say 40. There is a deletion. There's a deletion, and it's telling you that something has happened on this sequence. So that is sequence alignment. This is what we call pairwise sequence alignment. So I believe you grab that. If you are with me, are you with me?
Yes, sir.
Hello.
Yes.
Yes,
Okay.
sir.
Okay. Thank you. So this is pairwise sequence alignment. We have multiple sequence alignment, and as I told you that multiple sequence alignment, my favorite tool I use for it is Mega. And then the reason why we are not doing that this evening, the major reason why we are not doing that this evening is because I have not installed Mega on this laptop. So I was trying to install it yesterday, but I couldn't finish. So that's Mega. I use Mega for multiple sequence alignment, and then For multiple sequence alignment, I told you that we have so many nucleotides that you align together. What is the purpose of sequence alignment? Is just for you to look at the similarity. How is this sequence similar and all that? So at least I think by this time I have to end my section, and then before I end my section, there's still something I want to say. Now, Mr. Shen was talking about toxicity before you design your drug and all that. Yes, this is where bioinformatics come in. We have wet lab, dry lab. Bioinformatics is dry lab. You do everything. You do toxicity prediction, allergenicity. These are things that we offer at Great World Biotech Institute. So we have a cohort of students that we are actually training. I think the December last year we concluded a round of training for some of our students. Some of our students are still in training, so that's willing to advance their skills with bioinformatics. So if you are interested in joining the next cohort, is starting on Monday. This next week, or the next cohort is starting on Monday, and then with our seasoned bioinformatician, we'll walk you through everything. Believe me, these 2 hours workshop or webinar will not just you won't grab everything that has to do with bio. We can just lay our hands on some of those. So if you are interested in advancing this skill, you can reach out to us, our email, and any of our social media handle. If you are not on the general WhatsApp group, you can reach out to us, and then we have testimonies from of our students that will train on bioinformatics. So bioinformatics has come to stay, and then I will encourage you. I started the training during my third year in the university, and today we are still learning, and then I have others. Mr. Sheeran is a very good colleague. So when it comes to molecular docking, I give it to him. So and then as well, Mr. Faith, Mr. Damula, Specials. We have Miss Esther on the call as well. So as I've said earlier on, it's Great Obstacle Institute. If you are interested in advancing this skill, reach out to us. We have our materials, we have our international communities as well. We'll be able to train you and mentor you with skills in bioinformatics. Don't forget, bioinformatics is the fusion of biological science. So why can't you just start the journey now? So I'll be ending this call. If you have questions before we draw the curtain, you can please drop for any of the presenters. You can please drop your question now. Let's do that in the next 2 to 3 minutes before we end the call. So you can drop your question before we end the call. Thank you for staying put. Thank you for your question. Any question from the course of the presentation? You have your question. Any question from the course of the presentation? You have any question for any of the presenters where you need a little enlightenment? And I'm not seeing any question though. I believe we okay. Virtual screening and in silicon model. I think Mr. Shion will answer this. Mr. Shion, this is a question for a virtual screening and in silicon model. Mr. Shion.
Yeah, I'm with you. Okay, Mr. Femi, what was? Can I see the message again? Let me see. Okay, virtual screening and in silico model. I don't know. I don't really understand what it entails. They are just, I can say they are, kind of synonymous to each other. When you say virtual screening, as is more or less, we're having a virtual meeting now. So when you're screening compounds, protein targets, virtually. That means you're actually making use of a computer. You are doing that outside your immediate environment. And the in silico model is also talking about virtual screening, but this is the kind of standard way in which we refer to this process. We have in vivo and in vitro study of different diseases and many other things. So also we have in silico model. We can actually take up a particular disease and we look at some of the protein targets in it, and we screen different types of compounds around it and see if there is a potential compound that has not even been discovered yet, and we from that. So in silico and virtual screening is synonymous to each other. It's making use of computer technology to design one or two things when it has to do with diseases and that. So that's just it. I hope that answers your question, Mr. Femi. All right, thank you very much.
Okay, Mr. Shehu, we'll still have you back. For the answer section, for your section, please, you have to take that down in the next 3 minutes. If you can do that, please. You have the floor, Mr. Shehu.
Okay, I'll try as much as possible to manage my time. Please, I just need you to pay very prompt attention to this, so we can quickly do that. The other time, let me share my screen immediately so we can quickly do that. Let me share my entire screen so that we wouldn't have to keep going back. Okay, can we all see my screen now? I'm on the Google Meet page.
Yeah.
Can you see my? Okay. The other time I said when you pick up, please, Mr. Yevin, or somebody should mute. I'm hearing. So when you pick up a particular extract and you need to identify some of the bioactive compound, what you do is you take that particular leaf or anything, you take it and you just say bioactive compound of these plants, and you see some publications, you click on them, you search through. This is what we call literature search, not just assume. So you search. If you go through this one, in most times you usually find them in the I think this is a tab I opened before. Sorry, compounds. I'm trying to see if I can do that. So now just follow through compounds and beta leaf. Then when you search it, you have some. We opened this one the other time, but I told you don't just pick this flavonoid and we have different flavonoid, phenolic compounds, and many things like that. But you do a literature search. You usually find this in the look at this table. Bioactive compound isolated from Vannonia. I think that's the botanical name of bitter leaf. We have vanodalane and all these things like this. So you kind of copy out these. You don't, and if you are putting it into writing, you don't just copy. You cite the source as well. You copy it and put it somewhere. So this particular compound, I just want us to check it on a platform I called PubChem. Let me put that in the chat box so that we can copy it and explore it later. PubChem, this is the platform in which we have different compounds. The thousands or millions of compounds have been synthesized, and they are on this platform, PubChem. Then all you just do is copy and paste. Okay, this is what I copied initially. I go back to that place. I copy my vanodalane. I copy it and I paste it here. It's actually a compound. We never can tell. It can actually, you could see some interesting suggestions that pops up. Then you search it. Then you have informations around this particular compound. That person should mute. Sorry. So you click on the first one. This is what you search for, vanodalene. It's a compound, and you can see some other kind of similar compounds around it. So you click on vanodalene. So when you click on vanodalene, it will bring you to a page whereby you have more information about that particular compound. You could see the structure. We have the 2D structures, we have the 3D structure, we have the molecular weight, we have the structure, we have so many other information. We have the IUPAC name, we have the canonical smiles, we have the isomeric smiles, we have the molecular, formula, and we also, all these things. They have one or two things to do when it comes to designing a particular drug, checking for the toxicity of um, this particular compound, and we have thousands of them. Probably you work on a particular leaf. Pick up that leaf and let's see what we can do. I will, in the next 3 minutes, I want to quickly do something, a simple toxicity test, a simple one. So, like Mr. Yemi said, if you join the cohorts, there are other, kind of training we're going to, engage you with. But then let's have our hands on this. There's a particular um, website we call Protox. Yes, I'm on the page Protox. So we just use it to do simple toxicity test, and we from here we can actually come up with one or two initiatives so that at least we can learn one or two things today. So this is the home page. When you get to this home page like this, what you do is you click on the Protox prediction to want to check the toxicity of a particular compound. So we click on, there's a particular thing we call canonical smile. We retrieve that from this place. We just control C it, you copy it, then you come to this Protox and you paste it here. Then if you look at this place where my cursor is, this is where we have different types of toxicity we can actually do, and you get to know how toxic this compound is. We just run it, and if you are opportune to join the next cohort, you'll be able to learn the interpretation, how it applies and everything. So these are some of the toxicity tests we can do. Instead of visiting laboratory, beginning to isolate this and that, and you can conduct that. I've pasted the canonical smile here. I click on all this. I want to, kind of check the toxicity of this. Then I click on start tox prediction. Then I have my results with me. So these results will still need some other interpretations. You need to, then some other toxicity are still being calculated because some of these things will run online. A normal computer cannot run them. Like Mr. Amy said the other time, you submit a particular sequence to a database. If you submit it, if you try that with your system will just before lock up and never use like 5 hours to reboot. So we run some of these things on supercomputer and we access them through platforms like this, and we send our works to them and they help us to run it. You can see some of the results we already obtained now. These are some of the toxicity results of that particular compound, and we can make some derivative here. We can make some conclusions here, but time is no longer on our side, and as well, it's an opportunity to learn more about bioinformatics, computer-aided drug design, and so many other things as regards that. I would like to hand over now. I don't want to spend much time, like Mr. Yemi has said. So, okay, Protox. Yes. Okay, Protox. Let me provide the link. So, as a scientist, you don't just, the link is in the chat. You don't just come up with this kind of initiative and you just present this kind of result that, okay, this is you need to interpret this. You need to understand what everything entails, but then let's have a time we'll be able to explore and also explore other sources and other things as regards bioinformatics. Thank you, Mr. Amy. I hope I've not exceeded the time given to me.
Thank you very much, Mr. Singh. So, many more of these you will enjoy in our next course of our training and mentorship class. So, as Mr. Shona said, we have myriad of things you can do without your phone. I started learning bioinformatics is just a very small phone. So having a phone or not having a laptop is not a barrier, but if you have access to a very good and functioning laptop, that is very, very much good. So, for those that are interested, you can as well reach out to us. The next course is starting on Monday, and we are already on the course of training another young scientist. So, um, on this, we will draw the curtain here. I want to sincerely appreciate everyone that has joined the call and everyone that has contributed to the call. Miss Precious, our moderator, thank you. Time will not permit us of saying your bye bye. Mr. Shim, what a wonderful session with you. And for others, thank you, Mr. Ezekiel. Yeah, Mr. Ezekiel, thank you so very much, sir, for staying put. Your humility is top notch. Thank you. And for others, Mr. Femin is one of our students, though he has not finished his training. I should have said that publicly. So thank you, everyone that has joined the course. So we'll see next in our next course. So thank you, everyone. Do have a lovely evening ahead. Thank you, and God bless.